Hi everyone, welcome to Simple Funda. Today in this session, we are discussing about bacterial cell structure. This is the structure of bacterial cell. Bacterial cell shows prokaryotic structure which enclosed by three layers. The outermost covering is slime layer and middle one is cell wall and the innermost is cytoplasmic membrane. The slime layer is a gelatinous layer present on the outer surface of the cell wall, usually composed of polysaccharide or polypeptide chain of amino acid. When it is only made up of polysaccharide, it is called slime layer. If nitrogenous substance present along with polysaccharide, then it is called capsule. Cell wall. Cell wall is tough, rigid and granular. Three main constituents of cell wall are NAC, NAM and peptide chain of 4 or 5 amino acid. These are togetherly form polymer which is called peptidoglycan or mucopeptide. That NAC and NAM molecule are arranged alternately and run in one direction, whereas peptide chain run crosswise, which gives rigidity of bacterial cell wall. If the cell wall is removed by laboratory procedure, the naked protoplast becomes spherical. Cytoplasmic membrane. Cytoplasmic membrane is a bounding layer of the cytoplasm. It is semi-permeable in nature, which is about 75 angstrom thick. It is double layer of phospholipid molecule. Phospholipid molecule are of two types, that is hydrophobic and hydrophilic. The hydrophilic phospholipid present towards the outside and hydrophobic molecule towards the inner side. Cytoplasma and cytoplasmic inclusion. It contains fluid portion and storage particle and also chromatic or nuclear area and some other inclusions. But in cytoplasm, there is absent of mitochondria, endoplasmic reticulum, centrosome and Golgi bodies. Nuclear material. The characteristic feature of bacterial nucleus is the absence of nuclear membrane, nucleolus, chromatonema and nuclear sac. Such nucleus called nucleoid or genophore. That's why bacterial cell is called nucleoid. It is composed of double or single stranded DNA. It is usually ring-like structure and DNA molecule is approximately 1000 micrometer long. And it is devoid of histone. There is absent of histone. Plasmid. Plasmid is extra chromosomal hereditary determinant which are either independent of bacterial chromosome or integrated with them. Each plasmid carries non-essential gene and there has no role in viability of growth and bacteria. Ribosome. Ribosome are the site of protein synthesis. In case of prokaryote, there is 70S type present, but in case of eukaryote, there is 80S type of ribosome present. But in case of eukaryotic cell, ribosomes are present on the surface of the endoplasmic reticulum. But in case of prokaryotic cell, there is absence of endoplasmic reticulum, so that the ribosomes are free in the cytoplasm. That 70S ribosome has two subunits. The larger subunit is 50S and smaller subunit is 30S. Mesosome. The complex localized infolding of the cytoplasmic membrane are called mesosome. 2 to 4 mesosomes present in a cell due to absence of certain enzyme like ATPase, dehydrogenase and cytochrome. It is not the site of respiration, but it is suggests that it helps in respiration. But the main function of mesosome is form of transverse septum during cell division. Flagella. Flagella is a thin hair-like appendages and it is whip-like structure which helps locomotion in bacteria, pili or fimbriae. Pili is smaller than flagella. It is also hair-like structure. The length is approximately 2 micrometer and diameter 30 to 50 angstrom. Usually several hundred pili present in a bacterial cell wall. Pili are made up of special type of protein called pillin. Pili are not the organelles of locomotion. Its main function is adhesiveness. Bacteria are stick to one another with the help of sex pili and transfer the genetic material, usually 1 to 5 sex pili present per cell.